Okay, I'm back. It is Friday. Hopefully Thursday went well for everybody. Today what we're going to do is every class is going to go to the library today. Now I know first and third hour biology classes, you guys missed out on yesterday's activities, but I just want to give you a quick reminder that your bio babies and your chromosome drawings are due. Now I'm going to give you guys a little bit extra leeway because you were shorter today. So I'm going to let first and third hour turn their chromosome drawing, which was this assignment here. I'll let you guys turn that in on Monday. But your bio babies are due sometime today, and they are due on my desk or on Moodle sometime today. Now, if you're going to submit it on Moodle, you can go home and finish that. And if you have some sort of circumstance where you can't access the video, or I'm sorry, can't access Moodle, then you're going to have to let me know. But you guys have had plenty of time to prepare for that. So everybody knows about the bio baby. Now this chromosome drawing, that I'm going to give you a little leeway on if you are in first or third hour only. So bio babies are due today. You can turn them into the appropriate folder sometime today. Now obviously there won't be anybody in my room after 3 o'clock, so pretty much they're due by about 3:05 today. So that is the bio baby and the chromosome drawing. Now what you're going to do today, this is can be a little bit of a difficult assignment if you don't read the directions. So first thing you're going to do is you're going to go down to the library and you're going to go into Moodle and you're going to click on this assignment right here, the Sex-Linked Traits Virtual Lab. And then what you do, once it comes up, just like normal, you have to open this link in a new tab. And it should open up over here. Now, this particular assignment requires you to follow some directions. So if we scroll down, what we're going to do here is we're going to cross some fruit flies to look at a sex-linked trait. Now remember, sex-linked traits are ones where the gene for the trait, all right, the gene for the trait is on the X chromosome. And remember what is different about male and female. X chromosome, if there's 2 of them, it's a female. If there's 1 of them, it's a male, and the male would have the Y. There are no genes on the Y chromosome, and so that creates some unique inheritance patterns. It creates some unique results when we do the crosses or the Punnett squares. So how this lab is going to work is, first thing you're going to need to do is make this table down, create it in your notebook. Create this table in your notebook, and I'm going to allow you guys to work on this in groups if you wish. But this table here, and it's kind of long. You have to do everything twice, all right? But you have to copy this table down in your notebook. So that would be the first thing I would do once I got down to the computers. Now, at the end of it, you're going to have some journal questions here that you're going to need to submit into Moodle. And remember, since you're doing it as in a group, you need to put both people's names in the assignment where you submit it so I know who it belongs to, because otherwise I'm only going to see one name, and that's whoever's logged into Moodle. Now, what you're going to do, the procedure here is, these fruit flies, they're called Drosophila. That is part of their scientific name. All right, what you have to do is you have to click on either these jars or this notebook to do the crosses. I would recommend the jars, but you can do it either way. And what you're going to do is you're going to click on the jars. Now you have to decide who you want to mate. All right, which flies do you want to cross? So, for example, if I wanted to cross this fly right here, this is a red-eyed female. Eye color is a sex-linked trait in flies. So I put here like that, and there's my female. Now I want to decide which male I want to cross, either a red-eyed male or a white-eyed male. I'm just going to pick a white-eyed male for now. And you put them in there and you hit mate and sort. And now what it's going to do is it's going to give you some results. These are the babies of that cross. I have 49 red-eyed females, 51 red-eyed males. We have no white-eyed males and no white-eyed females. All right? So once you have done that first cross, then you go to your table and you fill in the appropriate data. All right? On your table, you're going to see P generation, you're going to see P1 cross. So in the P1 cross part, you put the parents, and all you have to put is the phenotypes. All right, so I'm gonna go back here and just bring that table up again, and show you what I'm talking about. So here's the table. And you won't be typing into this table, but I'm going to do it for you. So for the phenotype of the male, I think, if I remember right, I did a white male. Now for the phenotype of the female, I had a red eye. Put eyed here too. All right, so that's what we had for our P generation cross. All right, now what are the offspring? Number of red-eyed male offspring. And I don't remember what numbers I had, but I'm pretty sure I had only red-eyed babies. So I'm just going to make up these numbers, guys, so bear with me. But let's say I had, I don't know what I have, 48. Okay, there we go. 48 red-eyed males. I had no white-eyed males. I had, and I don't know what I had, 52 or something. 52 red-eyed female and no white-eyed female. So that's how the first cross looked. Okay? And that's how you fill in your table, just like that. Now what I'm going to do is I'm going to take two of these babies, one male baby and one female baby, and I'm going to cross them. All right? That is what we call the F generation cross, and then when I get the results, I'm going to fill those in. All right? Now you notice it starts back over here with P. What you have to do now is go back and do this whole thing again, do the cross again with two different kinds of parents to see what your results are. All right? Here and try that again. Oh, it's still there. Perfect. I was a little off, but these are the results. So now what I would do is I'd say, all right, I'm going to cross, since I only have two of these, I'm going to cross another red-eyed female here and red-eyed male from the children of the first cross. So this is the F cross. Hit mate and sort, and here are my results. Now this time it's different. All right, so something's up. This time I have 51 red-eyed females, 23 red-eyed males, and 26 white-eyed males. So that would go into my back here, but that was the F cross. All right, again, it was a red-eyed male, and it was a red-eyed female. Oh, we don't need to do that. All right, and then the offspring were a little bit different. I had, again, I don't remember what the number was. I had 50 something. And then I had, oh, wait a minute. No, I had something different, didn't I? I had 20, let's say, I don't know, 25. And I had 20 something. Again, I'm just making these numbers up. But then for the females, I think I had all I don't know, 48 or something. But I had no white-eyed females. All right? And then what you would have to do is you would go back and you would start over. All right? You would, let me see, I would reset this. Oh, there's my numbers right there, 51, 23, and 26. So that should have gone, no, I can't change this thing here. All right, well, I would reset it, and I would do all this again. Once I'm all done, I click on the journal questions, and I answer them in Moodle, where you hit your add submission down here. This is the teacher view, so it's a little bit different. Okay? So that's how. Now remember, if all of a sudden you click on these journal questions, 8 and it says undefined, all you have to do is refresh right there, and it will reload. Now it might restart you, but you should have your data already done, so it doesn't matter what's up here. Okay? So that is your assignment today, folks. Remember, bio babies are due today. Your chromosome drawings are due if you're in 4th, 5th, or 7th hour. 1st and 3rd hour, you guys have a little bit of a delay. You can turn that chromosome drawing in late. And then this, you need to finish up, and this is due by Monday, so you need to hurry and get it done. Okay? If you have any questions, again, please send me messages through Moodle or through email, and I will try to respond as soon as possible. Oh. And there we go. So I hope everything goes well today, and please contact me with any issues. Signing off.